And Ravi kindly asked me to come and speak to you about what I think about stem cells and how that relates to my Christian faith, and specifically the question of does religion hinder scientific progress? Now, this has been quite a real question for me, actually. My field, I'm not a stem cell biologist or embryologist per se. I work in the field of regenerative medicine. So regenerative medicine seeks to harness the potential of stem cells, to use this potential to regenerate tissue for transplant. You'll have all heard about the field. It's really, really well publicised, and it's a really, really exciting field to work in. And I've kind of bumped up against this question of, is it okay for me personally as a Christian to work with embryonic stem cells several times in my career? I've had several opportunities to form collaborations. I've been in various situations where I've been very entangled in this question. And so I've had to think about it in a way that's very real, not abstract, but in a way that's had to lead me to make real decisions that could potentially impact my career. But I'll come to my thinking on this later on. So my particular research is, I think, pretty cool. I'm trying to use hydrogels to deliver stem cells in the body and control their function. I'm working with a special type of gel that's formed from clay nanoparticles. Actually, the last time I was here in this building, I was giving a talk on this topic. Only about 3 people turned up to my lecture, so I'm really glad more people have turned up today. There was lunch for 20 though, so there's always a bonus. So I'm trying to use the interesting properties of clay to control the function of stem cells. Clay is a really sticky type of thing. It's these nanoparticles, and they're really, really absorptive for proteins and molecules. And so it's a very easy and convenient way of mixing in all the different kinds of biological signals that you can use to control stem cell function. And then you can see it's injectable, so you can inject it, say, into a hole in your leg or something with a load of stem cells, and the molecules within the gel and the stem cells then function to grow bone and fill up the hole in the leg. That's the idea anyway. So that's my research. And occasionally I've had the opportunity to work with embryonic stem cells. Routinely I tend to work with stem cells from the bone marrow because they're very good at forming bone and cartilage and fat tissue. That's the tissue I've been working with principally. But also there's the question of working with embryonic stem cells. So a bit of an overview of stem cell biology. Stem cells are a broad term that can encompass anything that any type of cell is able to proliferate and specialise into various different tissue types. I often view it as like a boulder on top of a mountain. Okay, at the top of a mountain, a boulder has 360 degrees of possibility open to it, and it's got loads and loads of potential. It can roll and roll and roll and roll. That represents an embryonic stem cell. The embryonic stem cell has the potential to form all of the different cell types that make up our adult bodies, and the reason we know that, of course, is because that's what we once were. We were once a tiny little ball of these stem cells, and these cells divide and divide and divided. They specialised and specialised until our bodies emerged over the course of development in all of their complexity. And this is what makes them so exciting and so interesting. If we can use this potential and harness it and direct it to grow, pancreatic beta cells or dopamine-producing neurons or something like that, we could treat all kinds of otherwise incurable diseases. And that's the excitement. But of course, the controversy is the source of these cells. In order to harness the potential of an embryonic stem cell, you need to extract an embryonic stem cell from a developing human embryo. It's taken at a very early 3-4 day stage. So this is a blastocyst, and embryonic cells are extracted from the inner cell mass. Now, these embryos are typically taken from people undergoing, from embryos that have been stored through the process of in vitro fertilization. So there's millions of these embryos in deep freeze, and they're viable still. You could take those embryos and implant them. Embryos survive very well in deep freeze, surprisingly much better than eggs or sperm, and you can implant them. So they're viable, but for embryonic stem cell research to occur, we need to take those embryos and destroy them in order to extract the stem cells. And this is where the controversy comes from. And it's probably one of the most prominent controversies in the whole of science. It's made famous especially by George Bush, So he took a very firm line on the issue of stem cell research in America, and he based his position very clearly on his religious beliefs. My position on these issues is shaped by deeply held beliefs. I believe human life is a sacred gift from our Creator. And this made massive controversy in America, and lots of scientists were very, very strongly opposed to President Bush's opinion and saw it as overly restrictive. This is Peter Singer talking on this issue. Peter Singer is probably the world's leading bioethicist, and he wrote an essay during that time, 2004, on the harm that religion does. And he says, if anyone tries to tell you that for all its quirks and irrationality, religion is harmless or even beneficial to society, remember those hundreds of millions of people. He's referring to the sheer potential that stem cell research could possibly have in treating diseases, who could be helped by research that is being restricted by religious beliefs. So he sees a very direct connection here between the harm of religion and the restriction that religion is putting on stem cell research. He goes on to say, and this is kind of indicative of the time he was speaking, meanwhile, just be glad that Christians in South Korea do not have the political clout that they have in America. Now this, in some ways, this is an argument of its time. This was a time when it all depended on opening up the restrictions on stem cell research and allowing a very important technology called therapeutic cloning. The problem with embryonic stem cells is that they belong to another person. These are cells that are from a developing embryo. They're not your cells. So in order to use them therapeutically, we need to be able to harness the potential whilst at the same time making sure they genetically match ourselves. And so a technology was developed called somatic cell nuclear transfer, and that technology was first pioneered and with great acclaim pronounced in South Korea by this chap here, Hwang Woo-suk. He claimed to have been able to produce 11 of these cloned embryos matched to humans, and he became an overnight star. He was flown business class all around the world, and this is what Peter Singer was referring to, because this was the big breakthrough of stem cell research of the time. But of course, you probably know this all turned very sour. Hwang Woo-suk turned out to be, first of all, very unethical. He was coercing his female postdocs and PhD students to donate their ovaries for his research. But also, he turned out to be a fraud, and he'd completely fabricated a whole load of his claimed clones, and the papers were retracted, and the stem cell field really took a big hit. But the other developments is not just this big problem that was encountered, and really that's still not been solved. But one of the reasons it's not been solved is because there's not much money being invested in it anymore. And the reason for that is because of another very important breakthrough by this chap here, Shinya Yamanaka, and John Gurdon, who's a Brit from Cambridge, Shinya Yamanaka in Japan. And they were able to generate these induced pluripotent stem cells. So pluripotency refers to the ability of stem cells to form all the different tissues in the body. And what this chap found is that you could basically take a fully specialised cell, so imagine our boulder rolled right down to the bottom of the mountain, it's specialised, it ends up in a specific location, making skin or bone or fat or something. You can take those fully specialised cells and turn on a few key genes and essentially reprogram them and then send those cells back up to the top of the mountain again. It's an astonishing discovery. And the claim was first of all made, and it was backed up by the key experiment in stem cell research, because obviously we don't know whether these cells can form a full human embryo, but what you can do is you can make chimeras with these cells. So you can take a mouse skin cell from an adult mouse, you can reprogram those cells, turn on those key genes that induce pluripotency, send it back to the top of the mountain, and this is what this picture's of actually. Those cells can then be injected back into the blastocyst. They then fuse with this developing embryo, and they go on to contribute to all of the different tissues that make this grown adult mouse. So it's an astonishing discovery. So it means that one day you can even recover these cells from your wee, old bladder cells washed out in your urine. So you can recover these cells from your wee, spin them down, turn on these genes, grow them up in culture, and then turn them into dopamine-producing neurons or something and use it to treat your Parkinson's. It's a revolutionary discovery because it means they're matched to you. So in some ways, this discovery kind of sidesteps a whole load of the ethical issues associated with stem cell research. But there's other things that are happening. In London in December, at UCL, there was a big conference examining the 14-day rule. The current UK restrictions prevent researchers from growing embryos in culture for any longer than 14 days, at which point they've got to be destroyed. And now a scientist in Cambridge and her group have shown that you can grow embryos right up to this 14-day period. And so the questions emerge, well, should we be able to go further? Should we be able to push this 14-day limit on? Now we can break the law, maybe we should change the law. And so there's a big debate at the moment, and you may have come across that in the newspapers. So the question of what is the status of the embryo, should we be doing research on the embryo, should we be destroying embryos, is very much alive. Now, I'm going to skip a few slides because I don't want to take too long. So let's examine some of the arguments. Now, I've said already that this is a real issue for me. It's also an issue that I've kind of changed my mind on a few times, actually. It's not, I think, an easy issue. It's not straightforward. And if it seems easy and straightforward to you, even James Thompson, the first person who derived embryos, said if you don't find this question problematic, you haven't thought deeply about it enough. If you don't find the idea of destroying an early human embryo troubling, then it might be that you're simply reflecting the beliefs of those around you and you're not thinking deeply enough about it. So I just want to spend a bit of time thinking about it. The main argument that's presented for pursuing stem cell research is always the sheer benefit that could come about by engaging in this research. So that's what Peter Singer says. Hundreds of millions could be helped by research that is being restricted by religious beliefs. This is the case that Obama made. Again, drawing on God, from a religious perspective. He said, as a person of faith, I believe we're called to care for each other and work to ease human suffering. I believe we have been given the capacity and will to pursue this research and the humanity and conscience to do so responsibly. So we've got to first of all consider this argument because it's the one that's always made. Think of the benefits that could result. Surely we've got to pursue this research because of the benefits. But of course you've got to be careful here. This is Elizabeth Anscombe, a philosopher in Oxford. And she reminds us that almost any field of medical research could put the claim that great advances might result from destructive experimentation on human subjects. And of course, this case has been made through history. The very basis of bioethics, of all kinds of medical ethics, and the basis for consent are those experiments that were conducted on human subjects. This is Carl Schneider, who was in the 1940s professor of psychiatry at Heidelberg University. He was talking about some research conducted by his colleague Julius Hallervorden, who was using the Nazi program of euthanasia to gain samples for his research. And he said this: the opportunity should not be lost to use the killing centres for research on mental diseases for their combat and in particular for their therapy and prevention. The problem with this utilitarian argument, this argument that says think about the benefits that might result from this research, is it can be used to justify all kinds of things. It can be used to justify great injustices. And so first of all, we've got to think, is this permissible research? Is this research on a human subject permissible? And so of course, the key question is this: what is an embryo? What is it? What is the human embryo? What status do we give it? What significance do we give it? An embryo sounds like a thing, doesn't it? It sounds like a kind of a thing, a type of thing. But actually it's not. It's a stage of something. An embryo is not a genus. It's not like a human or a mouse or an embryo. No, it's a stage of something. So it's better used as an adjective, actually. So you can talk about an embryonic mouse or an embryonic human. An embryonic something is a stage of something, like a neonate or a teenager or a child. An embryo is a stage of being a human being. It reminds us, of course, because when you look at it, it looks exactly the same as a mouse embryo. It looks exactly the same as a chimpanzee embryo, or whatever. They look very similar. So the very fact of our similarity with the embryo at the very beginning of our development reminds us that we are animals. We often use the word animal in distinction to humans. So Society for Prevention of Cruelty to Animals isn't interested in cruelty to humans. But we are, of course, animals. We are conceived, we grow, we eat, we rest, we defecate, we die. We reflect on those things. We're embarrassed when we're on the toilet. We prepare for our death. We celebrate births. There's some great distinctions, but at the same time, we are animals. And when you see ourselves at that very, very first stage of development, an embryonic human being, exactly the same looking as a murine embryo, it reminds us, first of all, that we're animals. We're part of the animal kingdom. But embryos principally are a stage of our development. Lots of people now take photos of themselves at different stages, more than ever before. I'm not sure what I think about that. My mum still brings out photos of me when I was a baby or when I was a child. And she says, and I can't remember anything about it. I can't remember any of my things she tells me about myself. But she says, that was you. That's what you looked like. And you did this and that then. And of course, if you killed me then, I'd have no memory of it. Well, of course I wouldn't, because I wouldn't be here. And you'd be doing an injustice, because you'd be taking away my future now. And when we kill an embryo, we're taking away a future. The embryo doesn't feel anything, it's got no awareness, but an injustice has still taken place. In the same way that killing me when I was a child would take away my future now, so killing me when I was an embryo, in the same way, kills me now, because there's a continuity. There's a continuity then between me then and who I am now. People also have photos of themselves nowadays. Those babies who came about through in vitro fertilization, their parents will often show them photos of them as a tiny embryo, as an 8-cell stage. And that reminds us again that the human blastocyst, even the human blastocyst in vitro, is a human stage. It is not humanly nothing. It possesses a power to become what everyone will agree is a human being. One could go even further. The human blastocyst is exactly what a human being is at that stage of development. Only its extracorporeal location is different. This is Robert Edwards, the founding father of in vitro fertilization, the scientist who was behind the birth of Louise Brown. And he even conceives this. He said, the last time I saw her, she was just 8 cells in a test tube. She was beautiful then, and she's still beautiful now. That fact of continuity is a very, very powerful fact indeed. I share my embryonic self and myself now share an identity which continues. And so an embryo is not humanly nothing. It is an embryonic human being. Now, so far, this is a very biological and slightly philosophical argument, mainly just biological. And so I think on the face of it, there must be a kind of prima facie case for treating the embryo as an early stage human being. But the next question is, why does this matter? Why does this human life matter? Now, there's different ways of answering the question why our lives matter. And there's a very definite Christian answer for why lives matter. There's alternative answers, though, as well. This is Peter Singer again. And he has a very robust, well-thought-out case for why life has to have only gained significance upon acquiring certain characteristics. He says, in his famous book, Practical Ethics, life only begins in the morally significant sense. So he makes a distinction there. This might be a human life, but it's not a morally significant human life. Because to be a morally significant human being, there must be an awareness of one's existence. Peter Singer sees that as the defining feature of being a significant human being, of having moral significance and worth. Now that sets the bar way too high for most of us. Most of us would say that's a problem, and the reason, of course, that's a problem is because it leads Peter Singer to justify killing infants. Killing infants who lack these characteristics cannot be equated with killing normal human beings. And again, he argues very strongly that infanticide is a legitimate option because infants lack self-awareness. And many of us feel, don't we, that for our lives to matter, for our lives to have weight and significance, we need to be able to do something. We need to achieve something. This is an unlikely jump of slides, I admit, but I'm just going to... This is Madonna. And here she is talking about her sense of significance and worth as a human being. And she says the reason for her drive, the reason why she has been so successful and why she's kept on going has always been to conquer some horrible feeling of inadequacy. And that's always pushing me, because even though I've become someone, I still have to prove that somebody. My struggle has never ended, and it probably never will. For many of us, we feel that we need to prove ourselves. We need to some way validate our existence as human beings. Even though other people might say we've got worth, even though legally we might have worth and dignity, really inside I can only find worth and dignity by achieving something, by doing something. I'm going to finish now. The Christian view of why a life matters is very different. And this is why the Christian struggles so much with embryonic stem cell research. Because the reason the embryo matters is exactly the same reason that I matter. Jesus, in a very striking story, kind of butts up against the value system of his day. Now they were bringing even infants to him that he might touch them. He's talking about Jesus. He is popular. People were bringing children to him. Infants are babies. And Luke kind of highlights the way infants were born. He says, even infants to him. And when the disciples saw it, they rebuked them. You see, in Jesus' day, across much of ancient history, infants had virtually no worth in and of themselves. Infanticide rates were very high. All of the great philosophers, Plato, Aristotle, advocated infanticide of especially disabled or feeble children, because that's how populations were made strong. That was basic. That was the value system. Infants had no worth at all. But Jesus called them to him, saying, "Let the children come to me, and do not hinder them, for to such belongs the kingdom of God. Truly I say to you, whoever does not receive the kingdom of God like a child shall not enter it." Now again, Jesus isn't talking about some kind of innocence or anything like that in children. He's talking about their emptiness, their vulnerability. And he's saying the reason these children have value is not because of any capacity they have in themselves, but because I care for them, I love them, and I welcome them. The reason you and I have value is because God knows us, he accepts us, he welcomes us, and he loves us. And for the Christian, that applies to me, and it applies also to this tiny life of the embryo. Last point. So, does this, therefore, inevitably hold back scientific progress? Surely it does. But let's just think about it again. This is a very helpful sentence by a guy called John Howard Yoder. He was a pacifist. He's writing about war, actually. He says, "I am less likely to look for a saving solution if I have told myself beforehand that there can be none, or have made advanced provision for an easy, brutal one." He's saying, if we've already accepted the fact we're going to war, then we're not going to look for alternatives. If we've already made advanced provision for an easy, brutal one, we're not going to exert our creativity and ingenuity to find a creative, saving solution. This was Shinya Yamanaka's motivation for doing his research, for making probably the biggest breakthrough in stem cell research, this breakthrough that won him the Nobel Prize. When I saw the embryo, I suddenly realised there was such a small difference between it and my daughter's. I thought, we can't keep destroying embryos for our research. There must be another way. By setting a limit to what he was willing to do, by saying, no, this is something we should not do, there must be another way, by kind of taking the scenic route, Shinya Yamanaka went on to make this great discovery. Sometimes limiting ourselves only means we exert our ingenuity further. It means we discover more things than we would have otherwise discovered. Thank you very much.